Hello guys, hello everyone. Apologies for the delay. So we are starting a bit later today because we were waiting for other people um, to join us, but we had some technical difficulties as it seems that the URL of the live streaming event has changed at the last minute. Apologies for that. And yeah, so hello everyone. For the ones that you don't know me or you don't know us, I'm Pablo Pito. I'm co-founder and CTO of Livebit here, and I'm here with Phil Palmer, our bioinformatician. And so today we wanted to give you a quick introduction about especially focusing on DeepVariant, a deep learning algorithm that was developed by Google, and talk a little bit about variant coding. So we are going to jump into the presentation. Okay, so yeah, so as maybe some of you will be already aware, so DeepVariant is a quite a good tool that was developed by DeepVariant by Google's team, Google AI team in this case. And so we are going to give us a quick introduction, and we are going to basically introducing you very quickly to variant coding, then to DeepVariant on how basically what are the main steps and processes of DeepVariant, and what we have been doing on using and being able to apply DeepVariant elsewhere. And then we're also going to show you quickly run through on how do we actually use DeepVariant kind of for a production kind of analysis for variant discovery and annotation. So as a quick introduction, so variant coding, many of you might be aware of, but for the ones that are not aware, so variant coding is actually the main step or the main kind of analysis that typically most of the genomics applications are looking forward, and it's mainly composed of three main steps. So in this step, the first one it's using just FASTQ files, and these FASTQ files, or the files that contains the raw data or the sequence data, can be coming from different types of experiments. This can be whole genome sequencing, but can also be exome sequencing, especially if you want to just focus on the coding regions of the genome. After the first step, the second step is getting the reads or these sequences mapped and properly aligned to your genome. And typically you have to use for that a reference genome assembly. And out of that, what you're going to be getting is BAM files. Right? And these BAM files can be compressed files that contain all these alignments for each of the sequence mapped into your reference genome. And the third and final step for detecting SNPs and mutations in general is the variant calling step, and that generates as an output a VCF file, which is the variant calling format file. So very much trying to simplify the workflow on how does it work. This is how it looks like. So first you start from the sequencing machine that returns you this raw data, FASTQ data, and after some kind of preliminary analysis and quality control, you run that through some kind of read mapper or read aligner. This one very common one is BWA, for example, which then returns you some file and you end up converting into BAM file with all your reads aligned. And then with those BAM files, with those reads aligned, that's when you can already start trying to call all these variants. And also typically what you end up doing, instead of every single time working from the FASTQ, so once you get into the BAM files or the alignment part, you are probably going to keep those alignments, and you can then afterwards keep using them in order to refine or rerun variant coding methods instead of just starting from scratch. And here we are showing you some pictures of the main formats from sequencing data. The first one is just the BAM file and how the alignments to the reference genome looks like, and the other one into the right bottom corner is the VCF format, where you can see what are the positions that you can find from your sample, from all your sequence experiments, and what are the mutations or the alleles in those cases that you can find in those samples. And after that, so there is also a very important data set that is typically used first in order to develop new methods, which is quite important, as if you are well aware about the development, the latest developments on the field, you will see that it's not extremely easy to get what's the best tool and what's the right method if you want to get the best out of your data. So in order to get a better sense of that, what researchers and teams tend to be using is a reference data set such as genome in a bottle. And that genome in a bottle, what basically defines is two main things. The first thing is ground truth using training data sets. And that actually defines across a set of samples and a set of sequencing technologies, just raw data that has been used in order to find variant. And since this is a reference data set or a ground truth data set for reference variant calling, what it also defines is the true, let's say, positives that you should be able, or that each method or tool should aim to find once you run any kind of analysis. And this reference data set is also actually quite well used in order to train different methods and to benchmark the results of any new tool in this case. So for variant calling or SNP genotyping in this case, there are lots of different applications, but the main applications and quite well known are discovery of disease-associated SNPs, also cancer cell monitoring or analysis, trying to look at mosaicism, especially in cancer cells, is actually quite interesting because of the wide range of mutation and the high mutation rate that cancer cells can have. It's actually one of the most complex scenarios for variant coding. Also pharmacogenomics and also population genetics and genome-wide association studies. So at this point, we would like to, I don't know if there is any answer, if there is any question that we might be able to answer. I think there is nothing. So otherwise, we will proceed right now. So right now we are going to introduce DeepVariant. So DeepVariant is an AI deep learning method that was developed and introduced by Google, a team from Google in this case, and is actually quite an interesting method because it uses pilot images. I'm not sure if you can see them here, but in the third column, for example, so after you do the read mapping, which is a prior step that you need in order to be able to train data or models in DeepVariant in this case. So after performing the read mapping and getting the alignments to a reference genome, so what you can get is actually literally snapshots or images, as these are called, these pilot images, that are literally all the reads aligned and stacked along your genome. And from those images, what you can capture, the information that you can capture, it's of course the alleles and the positions. And what this might be helping is actually to try to extract from all these images certain more complex patterns that will be otherwise quite difficult to extract with some other statistical model. And together in order to train the DeepVariant method or the models that are used within DeepVariant, you can use other kind of information. It uses quality scores from read data, also from the alignments as well, and you can add more features that might be related to the type of experiments or sequencing data that you are using in this case. And after training the models of DeepVariant, which is actually quite useful, is that it has been shown in the latest publications. It was initially published on bioRxiv quite some time ago, but now has been recently published in Nature Biotechnology. So it has been shown that actually after training DeepVariant models in such a large data sets, like can be genome in a bottle, for example, it has been shown that it can be actually extended. That model can be reused, not just for one reference genome, which can be HG19, for example, human reference assembly, but that exact model can also be used in other newer versions of human genomes, like on the latest versions. And it actually achieves quite a high accuracy in this case. and it can even be used. It can even be used with other kind of sequence technologies, and also even in some other species, for example. Even I think some of the analysis, DeepVariant managed to show that the accuracy of applying DeepVariant model training human data, when you try to call variants in the mouse genome, it's even higher than trying to train from scratch DeepVariant models from mouse data, which is quite impressive. Although it also kind of makes sense because of the large amount of and quality of the data that we have publicly available for human, right? And then also, so if you follow on Precision FDA, the Truth Challenge, there you have a really nice overview, quite a good overview of many variant caller, not just tools, but also kind of end-to-end workflows, starting from raw data up to variant annotation. And when this was actually published, initially it actually managed to achieve quite well results of accuracy in this challenge. And all that data is actually published, is publicly available in this case. And so I already mentioned, I think, some of the key features that DeepVariant I think that the DeepVariant actually provides. And one of them is that you don't need to do any kind of manual filtering along the process for variant calling. You get that straight away. And it's kind of an assumption-free, because as he said, you can train on a large data set, and that data set can actually learn a lot of dependencies in your data, and can be trained even for specific species. It can be trained for specific sequencing technologies, for example. And in this case is a convolutional neural network method. And and as I said, it's based on literally pilot images. And as I said, also it has been quite achieving quite robust accuracy results across different data sets of different technologies and across different species. And this is actually how these pilot images look like when you map all these reads across the genome. So on the x-axis would be a certain region across one chromosome. On the y-axis, you can see all those reads stacked in here. Okay, so if there are no questions, I think we'll continue discussing a little bit on how to actually try to run DeepVariant in this case. So we are going to be introducing a little bit what is our approach. Although of course you have all the information publicly available for DeepVariant, all the pieces of all the scripts and all the code and the model is publicly available. It has been published initially by Google, and you can find it out. But we are going to be presenting you what was our approach in order to put all the pieces together that were made available by Google, and how to be able to streamline DeepVariant and be able to combine it in any kind of analysis on a really nice and easy way. And for that we are using Nextflow as a workflow management language and system. And we'll also be presenting you a publicly now available pipeline that uses Nextflow and DeepVariant that has been published in the NF Core community. And then we'll show you, I guess, how this is run in production in this case. So I guess I'll head it to Phil Palmer, our bioinformaticians, that he will follow up with this part, presenting DeepVariant.
Okay, yeah, I can take over from here. Thank you very much, Pablo. Hello, everyone. So yeah, as Pablo was saying, you can see here, this is essentially what you need to run DeepVariant. And in dark blue here, you have all of the main steps of DeepVariant. And so these are all like binaries that need to be executed. And when we were trying to run this pipeline, we were trying to run it over deploy, it just really wasn't scalable, because there are all of these different steps you need to do, and you need to get all of the inputs from one, the outputs from one into the inputs for the next. And so this is where like Nextflow really came in. And so this was developed by Louisa, who worked here. And it's really useful because it takes care of all of the pre-processing as well. So super simple in that all you need to do is pass the BAM files and the FASTA file, which are in light blue here. And then everything else is taken care of for you, and you generate the VCF results at the end.
Phil, one question. So how did initially all these boxes that you have here, all these steps look like, and how did you have to put them together or to make them usable or runnable actually?
Okay, yeah. So they all run the like Google DeepVariant is already available on GitHub, and that's where you can find all the information for it. There's multiple ways to run it. One of them is through a Docker container. And then, as I'll talk about later, Nextflow has built-in support for Docker, which is super useful because then
so it makes you extremely easy to actually plug in all these containers developed by Google, right?
Yeah, exactly. Okay, yeah. And so then at this point you might be questioning, what is Nextflow? And so this here is Nextflow. This is super simple. It's like the kind of hello world of Nextflow. And at the top you can see the shebang line. So this is just where so then the computer knows to interpret this as a Nextflow script. And then below that you're using channels. So you're getting all of these different values, this bonjour, ciao, hello, hola, and that's all in one channel. And this is just how Nextflow passes data around. It's essentially just like a list. And then you execute the process, which is say hello, and here you define the input, which is this channel that's defined above. And then Nextflow will iterate through all of these different values in the channel, and it will echo hello world in all these different languages for you. And so one of the benefits of using Nextflow is you can have multiple processes. You can link them all together with different inputs and outputs, and they can even be written in different programming languages. So you can have one process written in Python, one in Bash, one in Ruby, and it doesn't matter, like Nextflow will know to interpret them all correctly as long as you use the shebang line for all of them. And yeah, that's a really useful benefit of using Nextflow. So Nextflow is a domain-specific language. So it's specifically used for bioinformatic analysis. And then some of the benefits of it are the fast prototyping. So it's essentially really, like, you can get a pipeline up and running really quickly. And especially do lots more like complex analysis than you'd be able to with like Bash scripts, for example. Like, while you would be able to do it, I'm sure it would just be a lot more difficult and a lot more time consuming to do so. And there's also task composition. So this is similar to the like processes. So each of them they can like each run in a different language, which is really beneficial. Especially if you're trying to kind of update like pre-existing pipelines, it can be really useful for that.
So I guess that's what makes it extremely easy, I think, for us, is right? Is because, as you said, if you suddenly receive something, you need to start working with something as complex as, in this case, DeepVariant. You need to be able to plug in any kind of software, right, that you want. And in this case, Nextflow allows you to actually plug in, put any kind of piece of code, any container, and compose this really even large and complex pipeline if you want, but on a really easy way without having to learn anything new, and just being able to reuse any piece of software that you have available. And I think that's what we ended up doing with DeepVariant in this case.
Yeah, and then the third benefit is also parallelization. And so because you're specifying all of the inputs and outputs, Nextflow can like model exactly what's needed and when, and so it can execute these in the most efficient way possible. And so you can have, like, for example, with DeepVariant, there's the make examples process, which is it's like the main kind of, it's the most computation intensive step, as we'll see later. And you can run multiple of them with loads of different BAM files all in parallel. So then rather than having to wait for 100 BAM files all to execute for make examples, you're actually just waiting for one and they're all running at the same time.
Okay, so you don't have to worry about the management basically of the parallelization and with as much data as you need, right? So that's just taken care of for you.
Yeah, and then as we mentioned, it's containerized. So this is really useful for all of your dependencies. You can use both Docker and Singularity. And it helps you rerun your analysis at any point. So it's a lot more reproducible, which is a really big problem in bioinformatics in general. So yeah. And then if you're looking to see where you can find DeepVariant, recently it's been released under NF Core. And so this is part of Nextflow. It's a community on GitHub of developers, and their pipelines adhere to these strict rules. So they're developing like multiple pipelines from loads of different companies. And if you want to release a NF Core pipeline, you have to adhere to like these 6 rules here, as well as like some other things as well. And so one thing is you have to have really good documentation. This is great just because it makes it really clear for anyone else trying to run the pipeline. And you also benefit because loads of different users and companies are all testing the same pipeline. So you can be pretty sure that it's going to be robust. And then the way that this is done is they have different like, config profiles. And so even if one person's executing it on a computing cluster and one person's running it on the cloud, you can all still run the same pipeline. Then there's also continuous integration testing, which is done with Travis. And so again, this helps just keep it more like stable and robust. And again, with the stable releases as well, this is so that all of the changes can be thoroughly tested before they're then released. And there's also the support not only of the Docker and Singularity for the software dependencies, but also Bioconda. And there's loads of packages available, and that can be really useful for installing almost any software dependency.
And it's actually also even really useful in the case of DeepVariant, right? Because that's what actually how we build all the pieces that use DeepVariant, or that DeepVariant is composed of. So we can actually build them extremely easily in the pipeline, combining Docker containers or Singularity, and together with Bioconda, where if you look at Bioconda repository, for example, you will also even be able to find the latest versions of DeepVariant.
Yeah, and so you can see here, these are all the companies that currently use and support NF Core, and LifeBit is one of them. And in terms of the pipelines that NF Core have, currently they've released 6 pipelines. So we're quite pleased that DeepVariant is one of them. And then they also have these 5 other pipelines, which I'd like thoroughly recommend testing. They're super great because they are so like easy to run and stable, and they're all very like similar in the way that they specify all of the parameters you need. And it's really good to have that uniformity across all of them. And so then in terms of DeepVariant, so I think Pablo's covered this already, but it was originally developed by Google, and you can run variant calling over DNA data. And so if you're looking to run DeepVariant, you can go to this link here, and you can see all of the documentation for DeepVariant here. And there's some information about the pipeline. And you can also look at the documentation, which can be found here. And then if you're interested in running the pipeline, then there's this section. And this lists all of the possible parameters that you might need. And so it's super useful. Like if you want to click on one and read any more detail, like here's all the different genomes, for example, that you can use. So these are already specified for you. And it handles all of the faster files and anything like that that you might need. And then this is the command to run. This is the command to run NF Core DeepVariant. And so as you can see, it's super simple. And
this is a simple command that anyone, as long as they have, in this case, what are the main requirements in this case to just have installed, in this case, Nextflow, and I guess Docker in your system, right? So just running that, it will allow you to run this DeepVariant pipeline straight away, even without having to clone or to pull this source code or the repository, right? You can just run it straight away in any kind of system, actually, which is quite nice. You can either run it on your laptop, you can run it in your HPC cluster, or whatever is the infrastructure that you have, right?
Yeah, exactly. So Nextflow has GitHub integration, so it pulls all the data you need for you, and all you need is to supply the BAM embed file, and then everything else is taken care of for you, and you can generate your VCF results. And then if that seemed like too much work and maybe you don't have Nextflow or you don't have Docker installed, then you can even run it through deploy, where it's even easier. And so I'll just show you how that can be done. So this is an example of one I did earlier today. And if you navigate to the pipelines page, and then you can see that DeepVariant is listed here. So if we click on that, and you can also see if you're not sure what parameters are needed. You can try with example data and parameters, or you can have a look at the documentation here. And so this lists some of the available inputs you can use. For example, what the only required input is this BAM folder. But what if you don't want to analyze all of the BAM files that you have in that folder? You can supply the BAM file prefix, and then it will only analyze that BAM file. So then if I go ahead and I'll submit a job, so I know we're going to need 4 parameters. One of them is the genome. So it uses HG19 by default, but just so that it's quicker to run, we're going to use chromosome 20, which is only used for testing purposes, really. And then to make it even quicker, we can run it with the exome model, and we can specify a bed file. So this is specifying the regions that you want to be analyzed. And so we can really subset it to just look at a very small region. And so here I've already imported the data, but if you'd want to do this, you can go click on import here. And then it's a public S3 bucket where we already have some of the data. And so here you can type in the name, and this is LifeBit featured datasets. And then I won't do this because I've already imported it, but yeah, the bucket is available here. And if anyone wants to sign up to deploy, you can do for free, and you get $10 of credit as well under LifeBit Cloud. And so then within that bucket, I'll show you, there's, I think this is the easiest way to do it. So in order to get the data, I'm looking in pipelines, and then we have DeepVariant here, and I'm using this quick test folder to get all of the data, which I've already imported here. And so using this folder, I'm getting the bed file, and using that for the bed option. And then I'm just going to change the BAM folder. Rather than running it on the example one they use, I'm going to use an even smaller BAM file, which is, so I need to select this folder here, and then it will look inside and grab the BAM file. And then click next and try. And here it's pre-selected the instance as well. So this is quite a large, powerful instance, which again will help the job run even quicker. And then again, run it. So now this has been scheduled. We can see here the status. So it's initializing. And you can also, now we can look at the one I've run already. And we can look at some of the analytics. And so we can see this is all the resources it used in terms of CPU and RAM. And we can even look at it by process and see how, as I was saying before, the make examples process is really the most computationally intensive, to the point where the call variance is actually far, far smaller than the make examples. And it was utilizing all of the CPUs of the machine. And then we can look at all of the processes that were run and see the pre-processing and then the main DeepVariant steps at the end here in order to generate the VCF. And then if we want to view the VCF, this one's still initializing, but it's be accessed here under the results. This is all of the output, which is produced by DeepVariant. And then we can click here. This is the final VCF, which can then be downloaded. And yeah, so that's how you would run DeepVariant. I can open the VCF file as well. And say, oops. Here is the VCF. It's just standard VCF version 4.2. And this is a list of all of the positions where there was a mutation. You can see what the alternate allele is and just where exactly this mutation has occurred. Yep, and then in terms of performance, we did some benchmarking previously. This was comparing bash scripts against resources optimized with deploy. So there's some resource optimization from deploy and also from Nextflow as well, because of the way that it parallelizes the processes. And you can see here, in terms of the cost across multiple instances, it was cheaper, sometimes far cheaper than using bash scripts. And you can also see here's the difference in runtime. And it was also far quicker to run as well. And then there's a new version of DeepVariant, so it should be even quicker now. And they managed to reduce the runtime by 65% from the previous version. So this is the version that NF Core is using, and we hope to implement it and to deploy it soon.
Yeah. So this last version is already included in the NF Core pipeline that is publicly available, right?
Yeah, exactly.
Yeah. Okay. So you can get, so if you get that pipeline straight away, then you will get the latest versions with the best performance. Nice.
Yeah. And then, are there any questions? So if there's no questions, then
yeah. So then I think we'll leave it we will, I guess, wish you Merry Christmas to all of you, and hope we can see you on the next year, on our next webinar. And for those of you who are mostly interested into kind of benchmarking and seeing performance, accuracy in general of DeepVariant, so for the next webinar, what we are going to be doing is having this fight between DeepVariant and GATK, where we are going to be showing how to run this kind of benchmark for a reference data set, and what are the results, so that you can choose or you can have a better informed decision on which one should you be choosing. And in this case, if you are interested at all in these kind of questions. So thanks to everyone for listening, and I hope you can now go and try it out, DeepVariant. And then of course don't hesitate to ask us any question if you need any information. We're going to be posting and sharing all the links and all the material in here. We'll make it available. But if you have any question, either regarding the DeepVariant itself, results, how to use it at all, how to use the NF Core pipeline, which of course, so Phil didn't mention, but he's actually the one, our contributor, the author of this DeepVariant pipeline in NF Core. So if you have any question, or if there is anything that doesn't work at all, then you have to stalk and to go after Phil in this case. And he will, no worries, he will answer straight away, no matter what's the time, what's the day. There is non-stop. It's non-stop Phil. And yeah, and of course, if you have also any issue, if you tried out deployed, just reach out to us if there is anything that we can help you with. Thanks, everyone.